Good evening. My name is Trishana Martin. Today's date is February 8, 2021. I am doing this results video for my microbiology class. It is Lab 6, biochemistry, biochemical, testing. I'm out of breath. Alrighty. So, and again, this is the results video if I didn't say that. So, I did get rid of a lot of the stuff, but I'll go ahead and just walk my way through it. We had to, let me get my gloves. We had to go ahead and incubate. I incubated for 48 hours, and I didn't see a difference in the broth of the E. coli and the S. epidermis. So I incubated another 24 hours, and you could start to see the bacterial growth. At that point, I had my, I'm just going to walk through my whole process. 4 tubes, 2 labeled E. coli, and the other 2 labeled S. epidermis. I separated the broths in each tube equally, and then from there on, in the first 2, E. coli and the epidermis, I added, I don't know exactly how many of the drops. I think it was 7 of each or 4. Can't remember. Added the drops in. In my opinion, immediately with the E. coli, I believe, the methanol drops it was a small layer, as you'll see on my pictures at the top. With the S. epi, it looked like it started to blend in the broth immediately. After a while, it stayed the same for the E. coli, but as far as the S. epi, it actually turned a reddish color. And that, I believe, means that it's positive. The other 2, we had to add the reagent. So a couple of drops of this reagent and also a couple of drops of the other reagent. This one is contains potassium hydroxide and everything like that. With those from the E. coli, I seen that looks still a little clearer. It had a cloudy, they looked the same almost. It just seemed like the S. epidermis was a little bit more cloudier to me. You'll see in my pictures, it's not too much of a difference, but if you look a little closer, just a tad difference. So that's the observation I made from that. And then we had to take our culture plates, and I do have those. I think I put them up. Bear with me, please. Oh. I keep them refrigerated because it helps me out a lot. I can show you these. So these have been incubating for a very long time. So as you can see, I did take some scrapes and I labeled my empty slide and scraped it onto the slide, the samples, and immediately when I did the E. coli, it bubbled up. The hydrogen peroxide, it bubbled immediately when I put it on there. And the bubbles stayed on there. They didn't fizzle down, it stayed on there. The same thing happened, actually bubbled a little bit more with the S. epi. When I put the drop of hydrogen peroxide, that one the same. It seemed like it bubbled a little bit extra. So those, the reaction to hydrogen peroxide, it was active. And so if I have to speak about what I would have done different, I think with this one, it was pretty much self-explanatory. It was a little bit easier than the other labs that I've done in the past, especially working with the microscope and calculating. This one seemed a little bit more matter of fact. So I don't think there's anything that I could have done. Maybe the incubation process a little sooner. But other than that, I think everything came out how it should have been. I got the responses that I was looking for. And so, again, how I always end my videos, I am very happy that it did. And I can't wait to see what the next week brings. Thank you guys so much for tuning in, and we will see you again.